So Oligon is a new company that will develop RNA therapeutics. We're working with CTI, and we have Paul here, CTI representing, and we're excited to set this up as a Canadian company. So let me jump right into it. Not sure how to move it forward, but yeah.
You're not exhausted, Mikey.
Oh, maybe that's, yeah.
Not working. Oh, maybe there.
Yeah, I'll do that.
Okay, there, yeah. Okay, there.
Thank you. So this is just a little overview of what I'll talk about today. So I'm going to start off by talking about the platform. What is the SeekR? It's a multi-targeted type of compound. It has aptamers on the ends, and it has siRNA in the middle. And I'll describe the technology itself and how we make these type of molecules. Then in the middle part of the talk, I'll introduce you to our therapeutic focus, which is overcoming resistance to cancer immunotherapy. And I'll talk about how this new platform gives us an opportunity to reprogram T-cells in vivo. And then finally, I'll talk about the lead therapeutic program and how we're planning to take that into the clinic. And I'll show you some data to support the preclinical data supporting how we're going to move that drug forward. So let me start with talking about RNA therapeutics in general. So they're different than small molecules or antibodies because those types of drugs need to be screened for and discovered and so forth. RNA, once you have a target, once you know the sequence of a target, and of course, next-gen sequencing allows us to know the sequence of almost everything now. We can now look at individual siRNA targets or combinations of targets, and we can target almost any gene in the genome. So there's no such thing as an undruggable target with digital oligos. Once we know what we want to target, we can synthesize that. It can be a small siRNA, it could be a large mRNA, but these types of drugs are digital in both their sequence and then how we make them. And you just literally punch in the computer sequence into a synthesizer, and you could use solid phase chemistry to synthesize any oligo of any length. And then the next step afterwards is you advance it into preclinical and clinical development. What's really unique about this opportunity in general is that it's not a 2 or 5 year, 10-year process. We could do this very rapidly. So you can go from an idea, a gene target, all the way to a therapy. So with COVID, a lot of people think it was a miracle that new mRNA vaccines could be developed within a year. Well, a lot of work went into the 10 years before the COVID vaccines came along, the mRNA vaccines, and recently the more successful RNAi drugs. So there's been about 5 approvals of drugs. Alnylam is one of the leaders in the field. But what I want to impress on you is because it's digital, you don't have to worry about cell-based manufacturing. You don't have to worry about antibodies. You don't have to worry about small molecule optimization and so forth. It allows you to really streamline the development of these types of drugs. So the state of the art now is the delivery. So lipid nanoparticles have been used to deliver siRNA, but the most successful strategy has been to link a binder to an siRNA. So this is an siRNA sequence here in the middle, and you can see there's a linker and then there's a binder. In this case, a receptor binder called GalNAc. It binds to the ASGRP receptor, GPR receptor. This binder is a small molecule binder that you can link to any siRNA, and then this naked molecule is self-delivering. So you don't need to put it into a lipid nanoparticle and so forth. So there's been one approval of this drug, but there have been all of these other 5 approvals with GalNAc, and they've been tremendously successful. And the risk in development has gone way down with these digital therapeutics. They have a limitation though. So GalNAc binds to a receptor on liver cells. So it's limited to hepatic delivery. If it's not a disease, it's not a disease target in the liver or in hepatocytes, then the GalNAc is not very useful in that regard. That said, we took the very same approach. We said, okay, what if we didn't use GalNAc, we used aptamer binders? And aptamers are like chemical antibodies. They're oligos, but they can bind and they can be made to bind to any cell surface receptor. That opens up the whole field. Now we can go beyond the liver to target any cell type, immune cells, brain cells, cardiac cells, epithelial cells. As long as you can find a specific receptor on a cell type, you can deliver what we call a seeker. So that's been our approach. And as I said, these are fully RNA oligo molecules. This rendering shows you that there's a tertiary structure at the end. So the single-stranded RNA folds on itself to form a tertiary structure. That chemical antibody then interacts with the cell surface receptor. So you have two binders on either end, and then you have an siRNA bridge in the middle, a double-stranded bridge that encodes for one or two siRNA or more. Okay, so that's the general platform, and I'll show you why it's been so successful. But generally speaking, these are self-delivering molecules. So you don't need to do anything other than make some chemical modifications to stabilize them in serum. But otherwise, there's no special formulation. You don't need lipid nanoparticles. And that makes a big difference because it makes the manufacturing a lot easier, but it also makes them a lot safer and a lot less toxicity than you would encounter with lipid nanoparticles. I mentioned that you don't need carriers, et cetera. These are injectable, so they can be delivered IV or subcutaneously. The other thing to mention here is that they're larger molecules than a typical aptamer or typical siRNA, so they go through less renal clearance. The glomerular filtration cutoff is about 30 to 50 kilodaltons. These molecules are typically larger than 50 kilodaltons, so we don't get the renal clearance. So we get better pharmacology, self-delivery, and most importantly, they can be directed to any specific cell type. As long as you can direct to any one or two cell surface receptors, you can get precise delivery, which is, again, a unique feature that we never had before. So how do they work? Well, these molecules, they once they're injected, they go through circulation. They'll find the receptor that binds to the aptamer, but then they cross-link two receptors. And they could be the same receptor or two different receptors, but that cross-linking between these two is the key. And the distance here is roughly the same. It's not shown there, but it's about 15 nanometers. That 15 nanometer gap is why you get cross-linking of receptors. So remember, receptors are, the antibodies. Antibodies are bivalent and they could bind to two receptors and cross-link. We get the exact same effect here because the distance is just right for cross-linking. Once you get cross-linking between two receptors, there's a very efficient receptor-mediated endocytosis. So this entire RNA molecule gets internalized into the cell, and the siRNA are then processed by dicer and the RISC complex to then go on and create RNAi, the gene silencing. So that's the secret sauce. That's why these molecules have been so successful and so efficient at internalization. And here you can see then the first generation of molecules, these chimeric molecules, have been developed by a lot of academics. So they've used aptamers as binders to siRNA. They work fairly well, but they don't have the pharmacology that you have with a dual binding because you bypass renal clearance. They don't have the efficiency of the bivalent cross-linking. So this next generation seekR platform has a lot of pharmacological advantages. It has better drug-like properties than we would get with that first generation molecule. And I'll show you some data to support that. Ultimately, once it gets in, it gets processed and releases the siRNA. And that RNAi effect could last for a month or more inside the cell. So if you want to reprogram a T cell, you treat it once, you can have a few doses over a week, et cetera. But then that RNAi effect, once it gets into the cell, it'll persist for about a month. Okay, that's what the platform is. Let me now switch gears and talk about how we actually make these molecules. So there's a three-step process. The first is you make the components. The first component is the aptamer binder. Remember, that's a chemical antibody, so it's a single oligo. And the way we discover these is that you go through a screening process. So you create random oligos, and the process is called selects. And what you do is you create random oligos with primers at the end. And that library is actually a vast library of oligodiversity. It's 10 to the 14 random oligos. That's a massive number, 10 to the 14. So you get an incredible variety of species of three-dimensional structures that can bind to any cell surface receptors. So then you go through about 12 rounds of positive and negative selection. You can use both recombinant protein or cell-based binding strategies. And through negative and positive selection, you find high affinity and high selectivity aptamer binders. That's the first step. Once you have that, then the next step is to develop the sRNA. And sRNA, now there's been over 20 years of development in terms of rules for how to select sRNA, make them more selective, et cetera. So that's a very straightforward process for developing specific and efficient sRNA. Some sRNA work better than others. Some genes are easier to silence. So there's a lot of variability there. But my team and I have been doing this since the year 1999. So we have a lot of experience with knowing how to design and make the sRNA. It's very straightforward. The next step is to then put those components together. So imagine you want to make a binder that binds CTLA4, and you want an sRNA that silences the STAT3 gene, which is exactly what we started with. You make the aptamer to CTLA4, which is a single oligo. So if you follow that oligo at the top there, you can see that this is actually a single chain, and then it folds on itself here. So this is the aptamer component. This is the antisense sRNA component. And then there's a single oligo here, which acts like a sticky bridge to anneal these things together. So it's really just 3 oligos, and you anneal those together. Very simple manufacturing process. What I'm leaving out is that you can manufacture these enzymatically, but you can also manufacture them using solid phase chemistry, which gives you a lot of precision and reproducibility in the manufacturing process. We also introduce chemical modifications. And I'll talk about that in another slide, but you can use 5' fluoro and 5' O-methyl chemical modifications that stabilize this molecule in serum, so you don't need to formulate it. But that's it. It's very straightforward to manufacture, a lot easier because it's so digital. You just enter the sequence in the computer, and then the solid phase chemistry synthesizer will just make it. It's a lot easier to manufacture than antibodies, where you get a lot of batch-to-batch variability because of the cell-based manufacturing. All right. So yeah, and this just shows you the specific oligonucleotide. So we manufacture these 3 oligos and then simply anneal them together, and the annealing protocol is very easy. That's the first generation. What if you wanted to make a bivalent dual binding aptamer, which we call a seekR? Well, that's the same thing. So instead of having just 3 oligos, now we have 6 oligos, and you can put linkers in the middle, or you can put sticky bridges. There's a lot of ways of engineering these. This is our favorite way to do it, which is to have 6 oligos. So you almost repeat the monomer into a dimer. So you create this first, and then you link them together using various types of linker chemistry. So that's how you make these molecules and how you manufacture them. I mentioned that there is a screening process. This is the selects process. Again, very straightforward. We use aptamer-bound aptamers, and one of the new tricks, one of the innovations that we've introduced into the field, and this is about a 30-year-old field, so we didn't invent selects, but we've improved on it because now we can use internalization as a way. So we care about not just affinity to binding, we care how efficiently they get internalized. So we use this as one of the positive selections. So when we harvest a cell, we can release what's bound on the outside and collect what's in the inside. And because this goes through an evolutionary step of 12 rounds of selection, what comes out at the end is highly selective. It's has high affinity, high selectivity, and it internalizes, which are the properties that we're looking for in this case. Once you know your sequence and you can use next-gen sequencing to sequence all of the oligos that come out of this, because again, you have primers at either end that allow you to characterize these things. Once you know the best binders, then you can synthesize these and you can further optimize them. Okay, so that's the platform. That's how we make these molecules. What are we going to use it for? You can use it for almost anything. You could target RNAi knockdown in any cell type, but we chose to start with T-cells for multiple reasons. One of the most important, though, is that T-cell biology is very amenable to this type of in vivo reprogramming, and we have cell surface receptors that we could target. But what's the end goal? So let me take a step back and give, and again, this audience doesn't need an introduction into T-cell biology, but just to mention that T-cells take various forms. So there's not just one T-cell, there's multiple subtypes of, and they're defined by the receptors on their cell surface and the cytokines they make. Just really simplify it for a second, and this is too simple, but let's just talk about effector cells and regulatory cells, two types. The top is our CD8 positive activators of T-cell biology, and then the bottom, the Treg, are CD4 positive, FOXP3 positive T regulatory cells. So you can think of the top one as the accelerator and the bottom one as the brakes, right? And these cells come from the same lineage, but then they differentiate to become different. How they differentiate, what controls their program is important, and it's really well known. And a lot of this biology actually came out of Toronto, right? So we know everything we know here about T-cell lineage and how they differentiate and what regulates them. It is really important when you're developing these types of therapeutic strategies. So we know, for example, that there are these master regulators, transcription factors and epigenetic factors that FOXP3, STAT3, NR4A1, ROR gamma T. These are transcription factors which are very hard to drug with small molecules. But with RNAi, we can knock them out individually or in combinations to try to reprogram these cell types. So that's the background, T-cell biology reprogramming. And again, think of T effectors as having more immunity, and the Tregs as suppressing immunity. So you have this yin and yang balance. And if you have too much hyperactivity of the immune system, again, this is oversimplification because there are other cell types like Th17 cells and other pathogenic T cells, but then you get autoimmunity if you have too much activity. And if you have too much suppression and you don't have enough activity, as you can see at the top here, if you have way too many regulatory cells and too few T effector cells, then you get in a situation which favors cancer. So tumors, for example, have what are called cold tumor microenvironments, and those cold tumor microenvironments favor less T effectors, more Tregs. Again, I'm simplifying, there's a lot of other cell types, but generally speaking, pharmacologically, what we want to do is we want to restore that balance and get normal immunity, whether we're trying to restore activity or suppress the activity in autoimmune disorders. So in pharmacologically, when you get too many, if you get pathogenic T17 cells and you get hyperactivation, you get autoimmunity, and you get much fewer Tregs. And then the opposite, you get, if you get immunosuppressive tumor microenvironment, you have a situation where you allow cancer to go unchecked. So let's talk about the cancer end of things. We know that checkpoint inhibitors, and again, CTLA4 is no stranger to this audience, right? TACMAC and team here have done tremendous work to delineate the checkpoint pathways and the intracellular pathways, but they only go so far. So PD-1 and CTLA4 have been incredible targets for immunotherapy. We know about 20 to 30% of certain cancers like melanoma and non-small cell lung cancer and certain other cancers respond well to PD-1 and CTLA4, but only about 20 to 30% of those patients benefit from checkpoint blockade with antibodies. We believe that there are intracellular resistance pathways, and I'm showing you one example here, NR4A1. NR4A1 is a transcription factor which gets activated as cells become exhausted. So the exhausted phenotype happens over chronic exposure to the same antigen. So think of cancer as the wound that never heals. So if you have that chronic inflammation, these cells eventually turn on NR4A1 and they turn on that exhausted phenotype. So the exhausted phenotype is characterized by upregulation of all these checkpoints. So LAG3, CTLA4, PD-1, all of those checkpoints get upregulated as these cells increase NR4A1. You get downregulation of IL-2 and interferon gamma and so forth, all of in the granzyme B. So they're not effective anymore in terms of their capability. This target was a few years ago. It was there's two Nature papers back to back that highlighted the importance of NR4A1 and T-cell exhaustion, and the references are at the bottom there. What really caught my attention is that how central it was. I mean, it's not just that it's upregulated with T-cell exhaustion. If you knock it out, you can actually reverse T-cell exhaustion. You reactivate those CD8 positive T effector cells, and you increase anti-cancer immunity. As seen on the right there, you can see that blue line. You get complete anti-cancer immunity restored simply by knocking down a single transcription factor. That's really exciting, because if this is what's holding back immunotherapy, the 70% of patients that are not responding, hitting that transcription factor, that's really important. Now, this one is hard to drug, though, because it's a transcription factor. We actually tried. We actually had a small molecule. We couldn't get it to, we couldn't get the chemistry to work because it's such a hard target. But we have an siRNA that works in picomolar IC50s. So this is easy for siRNA knockdowns. And now that we know how to get these in, we can hit the CTLA4 checkpoint with that antibody, with the chemical antibody that I showed you, the aptamer, and then we can hit targets like NR4A1, but it's not the only target. There's also STAT3, which is also one of these key targets. And this one's even more special because it doesn't just affect the T effectors that I talked about before. It also affects the Tregs. And remember, the Tregs are the ones that are putting the brakes. But if you have too much Tregs and they're all putting the brakes on, it doesn't matter if you have T effectors, they're going to suppress the immune effect. Turns out that STAT3 is important for both. If you knock out STAT3, it activates the exhausted CD8 positive effector cells, but it also suppresses and depletes the Tregs. That's excellent, because that's exactly what we want to do in the right direction for both cell types. Okay, so STAT3 has been known for a while, and again, it's got this yin and yang in terms of Tregs versus T effectors. It also has an effect in cancer cells, but I'm not going to talk about that now. But as a small molecule, there have been some small molecules developed against this. Unfortunately, though inhibiting STAT3 systemically in all cell types without directing it just to the right T cells has a problem. That you have hematological toxicity, GI issues, hepatotoxicity, neurotoxicity. This is not the right strategy for immune modulation. The right strategy is to get it just in the right cell types, the exhausted T cells that are overexpressing CTLA4, for example. We discovered that there was some really beautiful work that had been done at City of Hope. This work, and the reference is down here, this is using a first-generation molecule. It's just CTLA4 and an siRNA to STAT3. We subsequently made a bigger version of that, but this is all the data that I'll show you from City of Hope illustrates that these not only, well, I'll show you what the data shows in a second, but that was the first generation. The next generation lead IO that we're trying to bring into the clinic now has two CTLA4 aptamers and dual siRNA. And we've got two candidates, one that has STAT3, two STAT3s, and one that has STAT3 and NR4A1. And we're going to be developing this over the next year to move it forward into the clinic. So let me show you some data to prove that these molecules actually have the effect that I'm claiming. So first of all, let's look at, do they have the ability to get into T cells and knock down the target gene. In this case, the target gene is STAT3. So if we start with CD8 positive cells, and these cells typically express CTLA4, the more exhausted they are, the more CTLA4 they express, which is ideal for us. So you can see here, you can see at the top here, you get really good silencing of the mRNA. You can see it here too. And it localizes inside the cell. So it gets in and it silences the target, which supports the mechanism of action. That's in vitro. What about in vivo? When you treat mice with this SeekR, the tumor lymphocytes take up these molecules, and you can see here there's no treatment, and then the naive lymph nodes and then the tumor draining lymph nodes have a significant amount of uptake of these molecules, and you can see the silencing here of the mRNA in the tumor draining lymph nodes. So you can actually get knockdown of the target gene in vivo, which was another important validation of the mechanism. What about functional activity? Do these molecules, when you knock down STAT3, do you have an activation of the T cells? And the answer is yes. And you can use various markers. You can use granzyme B as an example. And you can see the black bars there. You get significant activation, interferon gamma, and other markers of activation. You can see, for example, PD-1 effects there. You can see that you can get down regulation of PD-1. Remember, as I mentioned, the more activated they are, the less checkpoints they have. So this is definitely activating the CD8 positive cell. And the most exciting aspect of this is not only are we activating the CTLA4 positive T effectors, we're also depleting the CTLA4 positive Tregs. And you can see here, especially in the figure above and the quantitation there, you get a dramatic depletion of CD4 positive, FOXP3 positive Tregs, dramatic depletion of those cells. And remember, those are the brakes. So we're accelerating immunity and we're removing the brakes at the same time. So the mechanism is really working in these. When you characterize the immune response, it's really beautiful and working in the right direction. And then you can actually see this also in other cell types, is in other models as well. So this is a model where we look at systemic treatment of B16 melanoma. Same thing, you see reduction of nodules and metastases. And of course, the most important takeaway here is not only do we have a very interesting immunological effect, we actually have an anti-cancer tumor effect as well. So the anti-cancer immunity is really terrific here. So let me walk you through the first slide here. So this is the B16 syngeneic mouse model. So there's a B16 is a melanoma mouse model. It's very resistant. So if you treated B16 with a CTLA4 antibody, or you treated with a PD-1 antibody, you don't see much effect. It's pretty resistant. If you treat with an aptamer to CTLA4 and then a negative control siRNA, in this case a luciferase siRNA, which doesn't hit any intracellular pathways, you see basically the same effect that you would see with a monoclonal antibody to CTLA4 or to PD-1. It's a very moderate anti-cancer immunity. If you inhibit the checkpoint and you knock down the intracellular pathway, you see terrific anti-cancer immunity, much better than you do if you're just hitting one pathway alone. And this is not the only, and this is, again, a very resistant, difficult to treat melanoma, just like the 70% of patients that are not responding. You also see it in renal cell carcinoma, lymphoma, colon carcinoma. In every case, you see incredibly good anti-cancer immunity just by knocking down these two pathways with, again, just the first generation compound.
What do you see if you wait for the mice longer time?
So what we're trying to do now is we're trying to get this flat line. So we want to get tumor regression. And to get that, we feel we have to get into more cells and more efficient delivery. And your question leads me directly into what we did next. And this is just to show that In T-cell lymphoma, it actually, we do get regression here. So to address what you brought up, what we want to do next then is to develop these larger molecules which are more efficient. Instead of getting, for example, 50 to 70% knockdown, we want to get 90 to 100% knockdown. And to do that, we need more efficient internalization. So hence now we're using that dual binding platform. So we manufacture these molecules, and this again, this is a collaborative work between City of Hope, where the first generation molecules came, and then Systems Oncology, which is an early asset incubator that developed over the last 2 years, developed the dual binding molecules which have better drug-like properties, which I'll show you in a second. So how much better have we? Oh yeah, and of course, there are many targets that we can put together in any combination. What I'm showing you is just the tip of the iceberg. STAT3 and NR4A1 are just the ones that have been more fully validated. The ones in bold are all ones that we have validated siRNA for, and we're working on some of these other ones. And Laszlo's blowing my mind with all sorts of new targets as well. He's got some ideas for other things we can combine there as well. So this is the original monomer. So it has one aptamer and one siRNA. Where we go next from this is we could put 2 of those together with a sticky bridge, and we can use chemical linkers or a sticky bridge. And now we have a larger molecule. This molecule had some stability issues. So when you put it into serum, it degrades. And the reason it degrades is this siRNA component here is unmodified. The color coding represents where we introduce 2' fluoro and 2' O-methyl chemical modifications. So you have some chemical modifications in the aptamer sequence, in the bridge sequence, but when you leave the siRNA alone, you don't see any. So this is tricky because the more you chemically modify the siRNA, the more you can mess up the RNAi effect. So you want to, you have the complex interacting with the RISC complex. You don't want to modify it too much, but we found just the right chemical modifications that gave us both stability and efficacy. So this is the final lead candidate that we were able to come up with. It's fully optimized now. You can see it's stable greater than 24 hours in serum. So the next step here then is to evaluate its ability to bind. So the dimer 1 and dimer 2 is the light blue is the less stable one versus the more stable and the more chemical modifications. You can see that not only did we improve the binding here, as you can see in this flow sorting, but we also improved the knockdown. So in an enriched population of CD4 positive cells, you can see we're getting down to almost 100% knockdown with dimer 2. Dimer 1 is actually pretty good as well. Look at the amount of knockdown we got with the monomer. And this is coming back to your question. This is the one that gave us pretty good anti-cancer immunity. We're hoping that with this new generation of molecule that we have now, we can get more sustained long-term activation of the immune response. And again, this is just showing how efficient this is, and it gets into CD4 positive effector cells. Just to reiterate the selectivity, we're very selective, not at getting into T cells in general, but getting into CTLA4 positive cells here, represented by these populations here. And again, it gets into both exhausted, stimulated, and exhausted CD8 positive cells. All right. So finally, the question is, well, did we improve the pharmacology by making a dual binding aptamer? And the answer is yeah. So if you compare the monomer, which is just one aptamer and one siRNA, to the dimer, which is 2 of each, you can see that the half-life in plasma went from 17 minutes to about 170 minutes, or 2.5. That's a 10-fold improvement in the exposure, and you can see the area under the curve here. So we get a 7 to 10-fold improvement in both the half-life and the exposure that we're getting. You can see the curves here. This is the monomer in blue, and then the dimer in red here. dramatic improvement in the drug-like properties. So next is we want to bring this molecule into further preclinical testing. And this is the one we want to drive into phase 1. Our focus in phase 1 is going to be 2 hypotheses. The first hypothesis is going to be in immune positive tumors such as melanoma and non-small cell lung cancer. But we also want to introduce some other solid tumors which typically don't respond to immunotherapy, such as colon cancer and other therapies in that regard. So that's where we are. I'm very excited to answer questions and discuss our plans in that regard further. Stop there. Thank you.